Excellent. Oh.
Hi, everyone.
Hi.
Hey.
Hi.
Hi, Alice.
All right. Good to see everyone.
Yeah, it's good to see that some people put on their cameras. It's nice to see who's out there.
Yeah, it's sometimes a bit otherwise like talking into the void, isn't it?
Yeah, that's very, very true. It's same with teaching. If you just present your lectures on a screen without knowing whether there are students listening.
Yeah, I know what you mean.
It's good that things are kind of back to normal in that respect.
Very good. I would say, yeah, we wait a couple of minutes because there are probably still some more people joining, and then we can get started. Yeah. We might also need to check whether we can set up a meeting where people automatically join, so we don't have to.
I have to press.
We'll figure that out.
Yeah. Hey, RJ, good to see you.
Good to see you too. Wasn't going to miss this.
Oh, that's good. Happy so many people are joining. Yeah, indeed, you have to admit everyone manually, right?
Yeah. We just kind of lost our Zoom license, so please apologies for these little technical glitches, but we'll get there.
What I'll do is, when we get started, I'll keep an eye on it and let people in.
Excellent. Thanks very much.
Latecomers.
That sounds good. Great. Oh, we already have around about 20 people. That's good to see. So maybe we can slowly start with the introduction. So I'm really happy to see Laura Govers from the Netherlands here today. I recently saw her presentation and some of her students' presentation at the ECSA conference in San Sebastian and thought, wow, this sounds like really interesting stuff. I'm pretty sure there are more people out there who couldn't join today, but would be interested to see this. So that's why we thought this would be a good idea to invite Laura to the ECSA Files. Hi, Martin. Good to see you. Could you maybe mute yourself? I'll hear myself. Yeah, thanks very much. All right, so Laura, welcome. A quick introduction. Laura is an assistant professor at the University of Groningen in the Netherlands, and also a tenure-track scientist at the Royal Netherlands Institute for Sea Research, the NIOS. And she studies conservation and restoration of foundation species in coastal ecosystems, and is aiming to advance the understanding of their ecological functioning to contribute to evidence-based marine conservation through a collaborative approach. That sounds really interesting, and I'm very much looking forward to hear more about it. So, Laura, would you mind sharing your presentation?
Yeah, well, thank you so much also for inviting me, Stephanie and John. So I'm very happy to be here, and I'll share my screen. So is this working?
Yes, looks good. Thanks very much.
Yeah, good.
Great. Well, thanks to all of you for being here, and thanks for having me. So today I would like to talk to you about how we actually approach restoration of altered seascapes, and also how we can actually link theory and practice to actually reach our goals in coastal restoration. So first, a bit of a general background. Yeah. So, well, of course, most of you are familiar with marine ecology, but as you know, most humans have always had the tendency to settle near our world's oceans. And this is actually an illustration from the Baltic Sea from somewhere in the Middle Ages that depicts the herring in the Baltic Sea. And the text that accompanies this illustration indicates that the herring in the Baltic was so dense that you could thrust an axe into their midst that would remain upright. And I think that really emphasizes how bountiful our seas actually were before we started over-exploiting them, but also how much people have always depended on the sea and our coastal areas. And not only for food, but of course, the sea has also provided us with a connectivity between terrestrial and marine communities, and also supporting human settlement near our ocean. So if you look at this map, you can see actually a lot of our mega cities in the world are actually built near coastal areas. So within 50 kilometers of the coast, and also like the mega cities that are projected to develop in the future all near our coastlines. So that has also affected massively our coastal landscapes. So we have not only throughout human history altered our terrestrial areas, but also our coastal seas. And unfortunately, our activities have also depleted, for instance, the marine megafauna, but also led to the global degradation of coastal habitats. But today I don't want to tell another depressing story because we're all familiar with that, but I actually want to provide a bit of a message of hope. Unfortunately, I think that the hope that ecological restoration and the restoration of our coastal ecosystem provides has also been acknowledged by the United Nations, which has actually declared this decade as a decade on ecosystem restoration. And I'm actually also emphasizing this because I think we as a community can also put a bit more effort in harnessing the full potential of this decade. So it's a great acknowledgement, but it doesn't really come with money. So I think that is a job that we have to do ourselves. But first, I would briefly want to discuss what we're talking about if we talk about ecosystem restoration. So the SER, the Society for Ecological Restoration, has defined ecological restoration as a process of assisting the recovery of an ecosystem that has been damaged, degraded, or destroyed. So it's actually really the practical aspect of ecological restoration. And the fields of ecological theory, restoration ecology, and ecological restoration are really tightly linked, where ecological theory provides us with concepts and predictive models to explain patterns and processes in ecosystems and also to identify causes of degradation. And ecological theory massively feeds into restoration ecology, which is the scientific process of developing theory to guide restoration and using restoration to advance ecology. And then we have ecological restoration, and I think it's important to make a distinction between these three, because ecological restoration is really the practice of restoring degraded ecosystems. But these fields are very tightly linked, and I'm actually very excited about ecological restoration, not just because we can provide tools and practice actually to restore degraded ecosystems, but also as scientists, I think this provides us with a unique opportunity actually to develop new ecological theory. Because actually, ecological restoration is not just about just understanding an ecosystem, measuring stuff, but actually putting it back together from scratch. And this is an example from a restoration project led by the University of Victoria, Canada, and the Galeano Conservancy Association, where they actually take students along in the field to really restore systems from scratch. And just by doing that, you learn so much about the functioning of an ecosystem. So restoration practice is actually our acid test of ecosystem understanding. I do hear myself echoing somewhere. So that's about ecological restoration, but what are we talking about when we talk about altered seascapes? So this is a photo of, the system I will focus on today, altered the Wadden Sea. Well, this is beautiful, but as I'm going to tell you later, it's not an unaltered seascape. And that's not just unique to this part of the world, but this map by Kriet Arenal actually shows that many of our coastlines worldwide have been altered compared to their original state, indicated in red and orange. So how have they been altered? Well, you can think about building dams and dikes. Well, of course, that's a very, very Dutch way of thinking about it. But also, for instance, in tropical areas by removing mangroves, seagrass beds, by removing salt marshes. So also by degrading natural systems, but also just by constructing on the coast. And today I want to emphasize on a system that I consider highly altered, but still it has a very high natural value. The Wadden Sea. So for those of you who are not familiar with the system, it's an inland coastal sea that stretches along the northern coast of the Netherlands, northern Germany, and the west of Denmark. And it actually contains the largest connected intertidal flat system in the world. It has a very high ecological value. It's very productive, and it's a very important stopover site for migratory wading birds on their migrational route between their wintering sites in West Africa and their breeding sites in the Arctic. And it has a high ecological value, but it has been changed massively. And also to understand how you approach restoration in such a context, I think it's important to understand how we have altered such an ecosystem. So for instance, we have built this massive dike, which is called the closure dam. That's also been depicted on this map. So this map actually shows the Western Wadden Sea in 1825 on the left side and on 2021 on the right side. And you can show that we built this big closure dam here, closing off a former estuary, the Zuiderzee, changing not only the ecology, but also the whole sediment dynamics and the hydrodynamics of this system. So that has actually affected the entire system in all its aspects. And it's not only that, despite its ecological value, there's still a lot of human activities ongoing, although it's a World Heritage Site. So there's ongoing dredging activities, gas mining, demersal fisheries, and also food web-wise, the Wadden Sea can be counted among one of the most overexploited coastal areas in the world, as has been shown by Heike Lotze in his paper in 2006. And there's also more challenges to come. So because it's such an important intertidal system, it's also very sensitive to sea level rise. And the latest models actually predict a minimum of 0.6 meter sea level rise in the coming century. So that's something, of course, an intertidal system is pretty sensitive to. And there's also heat waves. So this is a picture of a massive cockle die-off that we actually observed in last August after all these heat waves of the last summer. So the whole tidal flat was actually covered in dead cockles that were probably got overheated by all these heat waves. So there's a strong need for restoration in this system and also in other degraded coastal systems, mainly to promote biodiversity recovery, but of course also to promote the recovery of other ecological values and ecosystem services. And there's been several studies that actually have demonstrated that marine ecosystems really have a good capacity to rebound after degradation because they have a high adaptive capacity. So recovery may occur as a result of human efforts in restoration and conservation, which is also shown in this graph, a study by Duarte et al., that actually shows that many species have the possibility, if managed properly, to rebound. So that is definitely hopeful. But that also requires a massive effort on our side. So then the question actually is, if you take this into account, what do we restore a system to? And I think that's a very important concept that I want to touch upon in this talk. It's like we were just looking at how much the system has been altered, taking the Wadden Sea as an example. So what is your reference? What is your baseline? And we all know that shifting baselines are an important concept in fisheries science. So like, well, what we now perceive as normal as a reference is not the reference of our parents or grandparents. So this is an example from sawfish fisheries in Florida in the 1940s. Well, they look very plentiful, but now they're almost gone. So only people who were around at that time or looking at this photo may know that they were once plentiful. So, so that concept is very well known in fishery science, but I think that is also something that we need to consider in restoration science too. So what I want to touch upon is this reference concept for altered seascapes. So also, when you look at like the need for restoration, we can talk about shifting baselines. And here's an example of mussel beds in the Dutch Wadden Sea. So this is a study by Christiane et al. In 2015. That has actually attempted to estimate the area of intertidal mussel beds in the Dutch Wadden Sea since the 1980s. And if you look at this graph and we take the 1980s as a reference, you can see that the mussel beds heavily declined as a result of fisheries in the 1980s. And then they slowly started to recover. But of course, it's going slow. So if you use this reference, you would say, of course, well, there might be a need for restoration because we want to speed up recovery, for instance, and recovery is leveling off. However, there's another study in 2018, and if you look at the red line that depicts a mussel bed, that actually takes the year 2000 as a reference. And if you look at the year 2000, of course, our mussel beds now have recovered to some extent, even though its recovery is leveling off. So this is actually an example of, if you take two different references, you may actually draw a completely different conclusion about the need for restoration. And that is something that we need to consider. And I don't want to value like one study over the other. It's just like, this can also heavily affect the support that we get for restoration of these altered seascapes. So just considering this reference concept is really one of the most important pillars actually of ecological restoration. So ecological restoration needs to be effective, sustainable, engaging, but also informed by past and future. And that is a massive challenge, because when we work in these altered seascapes, a historical reference may not be valid. So then we have to think of something else. So that is, I think, just one of the major challenges that we're facing. Other challenges are, for instance, next to references, that there's high expectations. So we now have the decade on ecosystem restoration, but I would say that in marine restoration, we're years or decades behind on terrestrial restoration, which is a field that's about a century old, whereas we kind of started about 20 years ago. So we're actually doing well, but advancement is slow. We need proper science, but management expectations are high, so we need to manage that. And then in some areas, there may also be a perception mismatch, because people may see the sea as, or coastal areas as like pristine areas, because they're governed by tides and human impacts are not visible right away. That also affects the support for restoration. There may also be colliding interests of economic and ecological values. So for instance, fisheries. And I think one thing that we are also dealing with is, for instance, invisibility. Well, intertidal systems may still be visible to people, but as soon as you disappear below the surface, well, a lot of systems, people don't have connection to these systems. So the thing that we were struggling with is actually where to start if you have such an altered system. So, well, and I think one thing I really want to emphasize here is that I think the way to start it is actually doing teamwork. So in this talk, I actually also really want to acknowledge all the people who have contributed to this, have also influenced my work. So international collaborators, but also direct colleagues, lots of students, volunteers, all kinds of people have been involved. And I definitely want to recognize that. And also teamwork, I think, brings a lot of new scientific ideas and provides you with input to solve these challenges. Okay, so when we start restoring an altered seascape, we actually generally start with restoring habitat building species, such as ecosystem engineers, such as dune grasses. So these ecosystem engineers generally also affect not only their own species, but also their surroundings. And they can actually also modify their surroundings also to their own benefit. So a little refreshment for those of you who are not familiar with the concept. Species such as seagrasses, for instance, can attenuate currents and waves, and thereby they stimulate like settlement of fine sediment particles from the water column, thereby improving their own light conditions. So they can actually, in feedback with their environment, enhance their local growth conditions through these engineering properties. So that's an important thing to consider if you approach the restoration of these systems. And in the Waddenzee, we have two major ecosystem engineers, seagrasses such as eelgrass and dwarf eelgrass, Marina zosternotii, and shellfish reefs, in this case, Mytilus aedelis, blue mussels. So why do we start with restoring these systems? And that is because these systems usually provide the foundation of coastal food webs. So here's an example from a study we did in dwarf eelgrass beds in Mauritania, where we compared actually the food webs of bare intertidal flats, colonizing seagrass beds, and established seagrass beds. And all the nodes actually present individual species. And you can see that not only the number of nodes, so the number of species increases with like a succession of seagrasses, but also the number of links between the species increases. So these food webs actually become more robust in seagrass systems compared to bare intertidal flats. One kind of to visualize what that looked like. So this is something that we have developed for the project Biodiversity in the Dutch Waddenzee. So we want to restore these species, mussel beds and seagrasses actually to contribute to food web and biodiversity recovery. Okay, so I'm moving into like a bit more of an applied example from my own work in the Dutch Waddenzee. We have been attempting to restore seagrasses, intertidal seagrasses, in an adaptive manner, which means that we have been assessing our results every year and adapting our approach accordingly. And again, I want to acknowledge my collaborators in this project, which are also a nature management organization, Natuurmonumenten, and the Fieldwork Company. In the Waddenzee, we have 2 seagrass species in the intertidal, dwarf eelgrass and eelgrass. And we have mainly been focusing on restoring eelgrass in the Waddenzee, which is in the intertidal is an annual species. And this species, eelgrass, had historically also a very high cultural value. So this is a photo of guys in a suit and waders. I think very few of you would actually dress up in a suit when you go in the field, but they would probably. So this is a fleet of fishermen actually that were harvesting seagrass. In Dutch that was called wier. And they were harvesting the blades of subtidal eelgrass in late summer, harvesting the blades and then drying them and then using these leaves to actually stuff mattresses, pillows, but also as insulation material, or of course, even for building dikes. And that's also why the historical extent of subtidal eelgrass meadows in the Dutch Waddenzee was very well mapped, because it had an economic value. So in green, you can see the size of these historic meadows, and that was an estimated amount of 150 square kilometers. So that was really a proper amount of seagrass. Unfortunately, that has all disappeared. All this subtidal seagrass is now gone. And that has a number of reasons. So when they constructed this dike that I just showed, that is between, like, I don't know if you can see my pointer, the water became more turbid, and that actually coincided exactly with the wasting disease that actually wiped out a number of eelgrass populations in the Northern Hemisphere. Yeah, so and probably because of the light limitations, our eelgrass was really hit hard, and then within a few years in the 1930s, they all disappeared. And we also had, historically, we also already had intertidal seagrass, eelgrass, and dwarf eelgrass, but that has been poorly mapped because it did not have any economic value. But we do know the current extent, and that is actually very poor. So this is a map of the whole Dutch Waddenzee, and in these tiny green and blue dots actually indicate where seagrass still is. Well, that looks still like a proper amount, but all the sites have less than 1% cover. So ecologically speaking, you can say that they don't really contribute anything at the moment to the food web. So that's because so the eelgrass has disappeared because of this closure dam and the wasting disease, but also because of eutrophication. But there's also been a lack of recovery, and that's probably the combination of a lack of donor material available, because the northern German Waddenzee still has a lot of seagrass and it has recovered there. But there's, because the current is going from west to east, it's very hard for donor material to get to the Dutch Waddenzee. And we also still have a very high turbidity in the Waddenzee, also bottom disturbing fisheries, so many areas are not protected. There's a lot of bioturbation by lugworms. So there's a need for to aid the regeneration of seagrasses by restoration. And I think this study by Project Seagrass from the UK actually also shows that like if we act now, there's still a chance that we can actually recover our meadows, and not only our meadows, seagrass meadows, but also all the associated services that they provide. So we really need to act now and invest in it now to be able to regenerate our meadows. And actually, it's not something new, because seagrass restoration in the Waddenzee already started in the 1990s. So my colleague Marieke van Katwijk from Radboud University in Nijmegen actually already started to restore eelgrass meadows, or started small-scale experiments on restoration in the Waddenzee in the 1990s. And she mostly found that it was challenging and also especially to gain long-term results because it's such a dynamic area, but also that the results are very highly site-specific, and that it's really important to actually spread the risks. And in like the early 2000s, around 2010, the government, Rijkswaterstaat, the governmental agency, actually thought, oh, we have some knowledge. We actually just want to go ahead and do some large-scale restoration attempts, because in the US they developed this new method, so they thought that would be suitable for the Wadden Sea. So 4 years in a row, like large-scale restoration was attempted by actually harvesting seeds from donor material in northern German populations, so especially from Zult. Seed-bearing shoots were actually harvested and put in these mesh bags. So the BUTS is an abbreviation for buoy deployed seeding system. These seed-bearing shoots were put in this mesh bag, tied to these buoys and a rope, and then they would actually float up during high tide, like move around on the rope, and when the seeds would ripen, they would fall through the mesh on the ground, seeding a large area. So that was actually a very interesting method that had been applied in the US successfully and Chesapeake Bay, I think. And it actually looked pretty successful at the first glance. So in these red squares, you can actually see where these buoys were deployed. So that was the area seeded, and all the green squares indicate where plants were found in the next year. So this is monitoring data from Schiermonnikoog. So that looks pretty promising. So every square is 20 by 20 meters. So indeed, it's large-scale restoration. But if you look at the legend, that indicates that still every square only contains less than 1% cover. So that was still a seed loss of 99.99%. So something happened there, and that was, and also the successes were not sustainable. So it worked out for 1 year, but because we work with annual plants that are completely reliant on successful seed production, well, seed germination, plant growth, seed production, and then of course also seed retention. Like the year after this, there were only a few plants left, so it was not sustainable. So rather than doing large-scale restoration, we actually moved back to a more research-focused approach from 2014 onwards. And then we actually set also a number of clear restoration goals. So we aim to reduce our seed losses because, yeah, losing 99.99% of our seeds was just very inefficient, but also waste because we were collecting from natural donor meadows. But we also wanted to study the causes of seed losses and also find solutions to that. And well, linking back to ecological theory, we of course know that these species can display these positive feedbacks. So we actually wanted to improve plant densities to get these self-facilitating effects. So those were our goals, and we actually started by reducing seed losses. Well, we've looked into that, and actually we found out that our seeds were infected by a novel pathogen, a Phytophthora species, the water mold that is actually family to or related to the potato disease, terrestrial disease in potatoes. But it was a novel species, and it was also it had never been found in seagrasses before, and we discovered that our seeds had were massively infected by this Phytophthora species. So we estimated we lost about 40% of our seeds because it's a seed pathogen. Yeah, so that was highly problematic, and probably also because we put all these seeds together, we had such high infection rates. So we tried to solve that. So we developed the copper treatment method. So we were treating our seeds with copper sulfate, relatively low concentrations that are also used in organic agriculture to counter disinfection. It was very successful. But then, of course, we ran into another challenge, because you can't just throw copper in the sea. Yeah, so we needed to find a way to actually treat our seeds in a sustainable manner. So we moved on to indoor seed storage. So we still collected our seeds from our donor meadows in northern Germany. We processed everything, so separating the seed-bearing shoots from the seeds, and then storing them indoors throughout winter. So, and that was like, we could both treat them, but also that prevented seed loss through winter conditions like winter storms, etc. So we actually solved two problems at once. But then, of course, we ran into another challenge. So how do we get the seeds back into the field? Because we can't use this buds method for the seeds that were treated and that were all like separated from the seed-bearing shoots. First, we thought about hand broadcasting, but we tried that and that didn't work out at all because most of the seeds were just probably ended up in gullies and areas where they couldn't germinate. So we developed a new method, which we called dispenser injection seeding. It's a very elegant method. It's just a caulking gun, actually very simple. So it's an adapted caulking gun where we mix the seeds with mud from a local area, very dense mud. And then we can calibrate these guns so we can adapt the amount of seeds that we put in one caulking gun so that we actually know how many seeds we inject with every injection. And then we inject them into the sediment. It looks like this. So this one was one of the first years that we were injecting. So we have this little disc on the nozzle so that we can actually control injection depth. So actually a very simple method that turned out to be very effective. So actually in 2017, the first year that we trialed this method, we actually already had 100 times higher plant density than in 2015. So that was very promising just by using this method. But if you look at the plant densities, it's still very low. So only 1 plant per square meter, which is still not ecologically valuable. So we still felt like we needed to improve this method. So that's what we did. From 2017 to 2019, we were actually fine-tuning this method, trialing it at 2 different sites on Ginten and Uithuizen. And if you look at the photos below, we had a very steep increase actually in the number of plants that resulted from our method. And that was because we adapted a number of things such as injection density, injection depth, and the seed number per inject to optimize that. But also, like physically, we altered the method. So the tip of the nozzle is actually depicted in these pictures. So first we actually injected the seeds at the tip of the nozzle, but in 2019 we actually adapted that to inject sideways off the nozzle, so that if you pull out the caulking gun, that you wouldn't pull out the mud and the seeds. So and that was very effective. So actually we ended up being able to produce over 60 plants per square meter, so over 100% cover or 100% cover. And this is work that has mainly been done by my PhD student, Max Grefnings, who just handed in his PhD thesis yesterday. So that's great. So we finally actually found the method, and then we could actually trial other theories, right? So because if you remember from the start, these density-dependent feedbacks are really important, and that is something that we also wanted to test. So we actually also looked at the importance of planting design, and mostly at planting density and scale. So we actually seeded seagrass plots, eelgrass plots on 3 different scales: 4 square meter, 40 square meters, and 400 square meters, doing exactly the same things. And we actually found way bigger plants in our big scale plots, if you compare them to the smaller scale plots. And bigger plants actually also produced more seeds, so also yielding way more seeds that actually produce more plants in the next year. So we actually also found out that planting design is extremely important. And that is especially because we had scale-dependent water retention. So intertidal eelgrass is actually not super well adapted to growing in the intertidal. So it's very sensitive to desiccation. So our site actually is a very flat area near Ginten. And yeah, so a lot of water is retained there, and that benefits the eelgrass. But if it's present in very small areas, there's a lot of sediment entrapment, and the entire area actually raises itself. So what we actually had is large-scale self-facilitation, so water retention with all these plants, probably also leading to more fertilization because the pollen didn't have to move very far. But on a smaller scale, we actually found self-inhibition because the plants trapped so much sediment that they raised the whole plot out of the water, leading to more desiccation stress, but also because they trapped more organic matter, there was more sulfide accumulation, and so more sulfide toxicity. But actually, so we made all these, so we had a huge advancement in our methods and also in the underlying theory, but actually the coolest thing happened by accident. Because in 2018, the first time we started seeding here, so this is the Eind of Ghent in the middle of the Dutch Waddenzee, and the light gray areas are the tidal flats. So we started seeding at the red dot in 2018, but our method was not really perfect. So we lost a lot of our seeds that we seeded because they washed out, but they washed out to places where they could germinate. And then we accidentally started a new population. And the cool thing is that because these plants also produce seeds, the population expanded in the years to come. So in 2019, we had over 30,000 plants spread out over 100 hectares. In 2020, we had over 100,000 plants, also, of course, because we continued seeding, doing all these experiments. In 2021, we actually wanted to plant 1 hectare because we wanted to test whether our method was upscalable, which it was. So it was a massive effort of 250 people hours seeding 1 hectare with this method, but it worked out and we had beautiful weather. But this repeated seeding also helped out with to facilitate population expansion, so up to 270 hectares in 2021. And actually this year we had a crazy experience because the meadow actually kept expanding to over 650 hectares and over 1 million plants. So it's really, really taking off. And we had to conclude, we were very happy to conclude that actually intertidal eelgrass restoration is possible, which was something that has been doubted by a number of people, including a lot of nature managers and policymakers in the Netherlands. So this is a drone movie actually showing the aerial extent of our meadow. So all these green dots are our eelgrass plants. And so we started there from scratch. There was nothing, at least no seagrass growing there before 2018. And actually, I think we can say that we managed to restore the biggest intertidal meadow in the world, which you can now also partially see from space. So especially the dense areas you can see. And the cool thing also was that this also already led to positive biodiversity effects, which was, of course, also one of our project goals. So we compared the species richness of epi and endobenthic species in our restored meadow with the surrounding bare flats, and we actually found 32% more species in our restored meadow, especially grazers, but also mussels that attach themselves to the seagrasses. Yeah, so it's possible, and we think that we managed to do this because of teamwork and because so many people actually were involved in this, because we're constantly linking theory and practice using this adaptive approach. We managed to optimize our methods. And of course, it's also a matter of continuity. So we have been building also on the knowledge of previous projects, which is really important. However, unfortunately, so far, our successes have been location specific. So I don't know if you remember the picture of the DS method. So we also trialed that on another site, and there it was way less successful. So we still need to repeat our successes. And that's actually also why we are now moving towards like a new project that is funded actually by the government, which will still be seed-based. And we will start it, well, actually, we're starting it this year, but mostly next year, where we will focus also on dwarf eelgrass, because if you remember correctly, dwarf eelgrass is also present, and so far we've only focused on eelgrass, and we need to find other locations where we're going to repeat these successes and upscale the successes, and we're also moving our knowledge to restore seagrass in the Dutch Delta and Lake Gevelingen. And just one final note, I showed you this photo of, map of historical distribution of subtidal eelgrass, and I've only been talking about intertidal eelgrass, but I also have another PhD student, Katrien Reelmeijer, who's actually working on whether actually there are possibilities for subtidal eelgrass restoration in the Waddenzee, but we're really not far ahead in that there. And then since there's so many people listening, we're actually hiring a PhD student. So we're advertising for a position right now. So if you know good students, or if you're a student who might be interested in that, please go to the website of the University of Groningen and apply for this position. Well, thanks. And again, thanks to all the people who have contributed to this work. And thank you so much.
Thanks a lot, Laura. The virtual round of applause is coming in, I see.
Shall I stop sharing my screen?
Yeah, maybe for now. Maybe some of the questions we have also, refer back to slides, so then we can have another look. And I also see the first questions coming in. So we do have about 20 minutes left. If you have a question, please raise your hand and then we can talk about your question. So the first is from Mariana.
Hiya, sorry, I don't think my camera is working. I'm in the office and it doesn't seem to be working, but can you hear me okay?
Yes, we can.
No, hi, I really love your talk. I work with seagrass as well here in the UK. And I was just wondering if you've tried looking at anything like genetic variation in the donor meadows, just to see if you are dealing with mainly clones or if you have a lot of sexual reproduction happening, because maybe that could be an idea to avoid things like that mass infection that you had in the past if you had a little bit more variation in the donor seeds.
Yeah, so that's a good question. And interestingly, so we're only working with annuals. So there's only sexual reproduction occurring in our meadow, our donor meadow. So that's very interesting. So we don't have clones. So we know that actually the genetic diversity of this meadow is super high, and that's also known from previous work. But what we're mostly interested in is also actually looking at the genetic diversity of our established meadow, so of our new meadow. So we have been collecting samples for 3 years, but haven't analyzed them yet. So if you have any ideas for that, I'd be happy to talk to you about it. But it's interesting because we're working with annuals. We don't really have to consider like the problem with clones. And also the thing is, with this pathogen, what we found is that it's naturally occurring in natural meadows, also in our donor meadows, but there it doesn't seem to harm the seeds and the population very much, and probably also because we are putting everything together artificially for our for our restoration attempts. We're probably actually really facilitating this pathogen. So that's a problem there, but which we have managed to solve. Yeah.
Brilliant. Thank you very much. Yes, that's
welcome.
something I'm considering as well. I work mainly with blue carbon and carbon sequestration, but here at the University of Salford here in the UK, we work a lot with genetics. So we are trying to, we're coming up with some projects on seagrass and looking at these genetic variation in seeds for restoration as well. So yeah, happy to discuss and exchange some ideas in the future. Thank you very much.
Thank you.
Yeah, thanks for the question, Mariana. Next one is from Nigel. Oh, I see. Oh, there you are. You want to unmute yourself, or should we use the chat?
Sorry, are you referring to me, Nigel?
Yes. Oh,
Yes,
sorry.
great. No, I thought I was second. Firstly, thank you very much. We've been working on Noltii within the Salcombe Kingsbridge estuary in the southwest of England. I thought Marina was a perennial, so that's interesting in its own right. But the Waddensee is infamous for reefs of Pacific oysters, I believe, and we have a problem of Pacific oysters settling within our dwarf seagrasses. Do you have any solutions, please?
Oh, that's interesting. Yeah, just coming back to your Marina. So they can be perennial and annual. So perennials in the Waddensee occurred as subtidal. Like subtidally, they were growing perennially. Well, they had a perennial morphotype, and in the intertidal, they're annual. So because they really grow up on the same elevation level as Noltii. Going back to your, so we actually don't have, so our Noltii is not overlapping in terms of elevation or bathymetry with Pacific oysters. So that's interesting, but how is that problematic for you?
We're concerned that the oysters could eventually reef up and displace the seagrasses. Yeah,
I think they also have similar problems in Arcachon Bay in France. Well, where they have where they're actually growing Pacific oysters for oyster culture, and so there's areas actually where they have to remove the oysters also for seagrass. But I think they can actually also benefit Noltii if you want to restore them, because they can provide protection, right, shelter for Noltii to settle behind. I don't have any real solutions for you. I'm sorry, but I'd be happy to discuss this because I think it's interesting. And I know, for instance, also that, well, similar things, but are occurring in, I think, the Pacific Northwest. So the North American West Coast has Zostra japonica, which is very similar to Zostra noltii, also where Japanese oysters are also coming in and they're growing together. But I'm not sure whether they're displacing each other. It also depends on your system probably, and also like how much fine sediment the oysters are actually trapping and whether they can that fine sediment can suffocate or lead to more sulfite stress in dwarf eelgrass. But I'd be happy to discuss that another time. Yeah.
Yeah. Okay, I'll make contact. Thank you.
Yeah, you're welcome.
All right, thanks, Nigel. Next question is from Mike Elliott.
Hi there. Hi, everyone. Afternoon. Hi, Laura. That was a great talk. That was really nice. Can I go back to the loss of the seagrass beds with the Aslophdyke being produced, the Zydus E being closed? Because intuitively, when we started looking at restoration, and we were talking about, you might have remembered, we were talking about type A and type B eco-engineering. You know, type A where you try and recreate the right physical conditions, and then type B, you have to start artificially bringing things in and reseeding. And isn't there the danger that because of the Aslophdyke, it's almost difficult, it's almost impossible to recreate the hydrographic conditions that there used to be? Although actually also intuitively you would have thought by creating a more enclosed bay that you have done, where it doesn't have as great a scouring and all the rest of it, it might be better conditions for seagrass than it was before. But say this central point about whether or not you've created the right eco-hydrology in order to get the things developing once you've started.
Yeah. Hi, Mike. Thanks.
Hi,
Great question. Yeah, good to
nice
see
to
you
talk to
too.
you.
Yeah, so I think that's very interesting and something I haven't mentioned, that of course before you attempt restoration you actually need to make sure that the abiotic conditions are suitable, right? And that is, I think, also something that is questionable for parts of the Wadden Sea right now, because we have imposed some irreversible changes on the system, and still, for instance, the sediment is not yet balanced out. It's almost been 100 years. So, but interestingly, it works on one of our sites, right? So it's not that the whole system is unsuitable, but probably the suitable sites are way more limited. So we have also been, well, not me, but some colleagues have been making these habitat suitability models, actually kind of deviating from historical references, but more looking at what are the current habitat requirements for these species and where are those suitable areas in the Wadden Sea. So they have been creating these habitat suitability models, and according to these models, about 10% of our tidal flats should be suitable for eelgrass or dwarf eelgrass or a mixture. But so far, we've actually also, I haven't mentioned that here, like some of the most suitable sites, we have also attempted the method, because first we thought, well, we don't have a suitable method. Now we do.
Yeah.
But those sites did not appear to be as suitable as the model predicted. So the application of these models in real life is still challenging, also because it's like all these fine scale things that are occurring are not included in the models. So
I think that's one of the important points, that we've seen so many restoration schemes around the world which they work on a small scale, but when you try and upscale them, when you try and recover some of the areas that we're talking about, they just don't work. You know, you've got so many other influencing factors, sort of thing. So models of restoration, models. No, they're good for some things. It is how we and this is the big challenge of the decade of ecosystem restoration, that when we're writing about that, we can do lots of these small scale studies, but actually getting them to be, let's call them ecologically relevant. I mean, some of you have heard me use the phrase good for the ecologists rather than good for the ecology. You know, they make us feel as though we're doing something, but actually they don't have a big enough effect. But that's why we need more background studies like yours, which is good. So
Yeah,
That's
what I personally think is like, of course, we want to repeat those successes, but if you can kickstart a really big population, then also you have the donor material that can spread naturally, and then like you can actually colonize way more places than you could actually do artificially just or manually.
Yeah.
And I think that might also be a way to actually restore a core system, and then it can distribute or disperse from there.
Yeah.
Yeah.
Good stuff. Thanks.
Cool, thanks.
Yeah, thanks for the question, Mike. Do we have some more questions for Laura? If not, I'm sure. Oh yeah, Alice has a question.
Yeah, thank you for that. It was really interesting. You mentioned earlier about the kind of high expectations around restoration projects. And my question is kind of, how do you get from that small scale trial to the large scale and the funding, and how do you kind of actually get people to buy into it and show that it might not happen overnight, but it's a long-term thing? Like, have you got any kind of advice for that?
Yeah, I think that's also why it's so important that we share our stories, because I think it's a big challenge. And like if we share the success stories that actually show that just like if you have a bit of patience, it may pay off in the end. Because of course, like this is not something we could have achieved without like all the work from 20 years past, right? So we're just building upon these successes. And I think that is an important message to convey, and also to be very clear about those expectations. Like, okay, we're attempting it. And I think also as a community, we're getting better at this, right? So actually we have more success stories to share, which is very encouraging and also needed. But of course, and I think also we should perhaps, I mean, of course, I'm also just reporting our successes, but there have been a lot of very discouraging failures in this story too, right? Which we learn from. And I think that is an important message as well, is that a failure is not a failure if you measure why it failed and can act accordingly, like to adapt and do like adapt your strategy. And I think that is important for us as a community to communicate that to practitioners and nature managers. It's like, and we're all super passionate about this and very highly motivated to make it work, but we're not magicians, so so it takes time, right? Yeah.
We have a question in the chat from Will Manning. Can you see the chat, Laura?
Yeah, I can see it. So Will is asking like a quick question about the caulking gun technique. You mentioned burial depth changes. How important has burial been? Trade-off between loss of dispersal rate predation over burial losses. So actually this burial depth or the injection depth has been extremely important actually to make our method site specific. So also before attempting this method at another site, I would definitely also like do a small pilot experiment where you change this depth, this burial depth, because that actually allows you to compensate for natural erosion sedimentation dynamics on a site, especially if you don't really have proper knowledge about that, because these seeds of both Multi and Marina are so sensitive to burial, because the seedlings are just really tiny and they can't really grow, outgrow more than 3 centimeters burial depth. But of course, you have to kind of compensate for the natural sediment movement of a system. And of course, that's also why you should not attempt this method in a system or seed-based restoration of these species in a method with high sediment dynamics. So yeah, so for us, it's been mostly about the burial depth, about natural sediment dynamics, and not so much for predation losses, because actually the one site that didn't work out for us, that was due to predation. Actually burying them also exposed them to predation by ragworms, common ragworms. They were eating the sprouting seeds, so they completely prevented seedling settlement there. So it's mostly to compensate for natural sedimentation, and I think that is extremely important, especially in intertidal systems, which are highly dynamic.
Thanks, Laura. Cheers.
Thanks.
Great, thanks for the question, Will. Not sure. I also just posted the PhD project in the chat. If people are interested, maybe check it out. It sounds. I already have a PhD. Otherwise, I would say, "Oh, that sounds good." But yeah, maybe people know someone who is interested and hasn't had a PhD yet. Excellent. Well, then, thanks again, Laura. Thanks everyone for joining and the interesting discussions. Before we finish, the next ECSA Files is, I just had a look, on the 1st of December. Jonathan, could you remind me who we invited?
Yeah, so our next talk is going to be given by Salome Vincent, who will be talking about anaerobic microbial diversity in tropical coastal sediments. So that'll be on the 1st of December.
Great. Looking forward to that. As always, if you have not signed up for the sort of regular invitation, then we'll share the link to the registration form again, right?
Yes, yeah, to the normal channels.
Excellent. Good. Then, yeah, thanks again, Laura. Final round
Yeah,
of
you're
applause.
welcome.
And thanks everyone for joining, and see you in December. Bye.
Thanks, everyone.
Bye.
Bye then. Bye.